Hello everyone, my name is Schiavon, and in this video I'll be presenting a specific breeding method used in genetic improvement plans in Oryza sativa, more commonly known as Asian rice. So the first modern work in genetics began with a man called Gregor Mendel, who is now widely regarded as the father of genetics. Mendel conducted experiments throughout 1856 and 1863, publishing his work and findings in 1866. Mendel's work highlighted the fundamental laws of inheritance and how traits can be either dominant or recessive, and how they can be used to predict certain traits of another organism. Mendel created 3 laws of heredity. First law is the law of segregation, which states that in organisms that sexually reproduce with meiosis have 2 copies of every gene, and only one of the genes is passed to the next generation. The second law is the law of independent assortment, stating that genes for different traits are sorted separately from one another, so that the inheritance of one trait is not dependent on the inheritance of another. The third law is the law of dominance, stating that an organism with alternate forms of a gene will express the form that is dominant. Genetics is extremely complex. Trying to produce a plant with all desirable traits using conventional methods such as selection, hybridization, polyploidy, and induced mutation is effective but also time-consuming and costly. Genetic engineering is a method of direct and manual manipulation of genetics, altering the DNA in an organism's genome. Altering DNA can be done by introducing new genes, deleting DNA, or by changing a base pair. It can be used to extract DNA from one organism and combine it with the DNA of another individual. Desirable traits that Asian rice plants are bred for include grain yield, disease resistance, culm length, pre-harvest sprouting resistance, eating quality, grain weight, grain chalkiness, and grain shape. Particle bombardment is a technique used in genetic engineering designed in 1987 by Professor Stanford and his co-workers. The technique is used for plants that do not respond well to the agrobacterium gene transfer technique. Particle bombardment, also called ballistic particle delivery system, or a gene gun, is a device that fires pellets of heavy metals coated with DNA, with the objective of transferring foreign DNA into plant cells or tissue. Specific genes and traits can be introduced to plant tissues and cells through the use of this technique. The gene gun needs to be calibrated specifically for each type of tissue it's being used on. In this table, the calibration is for the gene gun is to be used on basmati indica rice. The top section here is a helium pressure gas chamber, which at a certain pressure will break a rupture disc, which is positioned inside here, firing into this vacuum chamber here. Shockwaves generated by the rupture disc will be fired into DNA coated particles, which are stationed inside this disc here. The stopping screen here allows the micropellets to pass through and transport the DNA to the target tissue, plant tissue, which is located inside this dish here. The particle bombardment has an advantage over other techniques and methods against transformation-resistant crops, such as rice, corn, sorghum, chickpea, and pigeon pea, as it is a direct input of the DNA into the tissue. The technique is also useful as it can deliver DNA to a large number of cells easily and rapidly. Disadvantage of particle bombardment are that it can cause cells to be damaged or ruptured from the shock waves. It is not a specific method of transportation, and as such specific cells cannot be targeted for DNA transfer. Genetic manipulation through conventional methods can be extremely time-consuming. The use of genetic engineering techniques such as particle bombardment are reliable and fast techniques that can be used in gene manipulation. Particle bombardment is an effective technique to be used where agrobacterium gene transfer may not be effective. Thanks for listening.